Hello students, welcome back. In today's session, let us learn about signal transduction via G protein linked receptors. So GPCRs or G protein coupled receptors are also known as 7-pass transmembrane domain receptors, 7-TM receptors, heptahelical receptors, serpentine receptors, G protein linked receptors. So basically, these are a large superfamily of cell surface signaling proteins that bind extracellular ligands and transduce signals into cells via heterotrimeric G proteins. So coupling with G proteins, they are called 7 transmembrane receptors because they pass through the membrane 7 times. Here is the image of a G protein receptor. So if you count the number of domains, it has 7. 1, 2, 3, 4, 5, 6, 7. Okay, so they pass through the membrane 7 times. So that is the reason why they are called as 7TM or 7 transmembrane receptors. Ligands can bind either to the extracellular N-terminus and loops or to the binding site within the transmembrane helices. For example, rhodopsin-like family. They're all activated by agonists, although a spontaneous auto-activation of an empty receptor can also be observed. So G protein coupled receptors are found only in eukaryotes, including yeast, cyanoflagellates, and animals. So the ligands that bind and activate these receptors include light-sensitive compounds, odors, pheromones, hormones, neurotransmitter, and they vary in size from small molecules to peptides to larger proteins. So there are two principal signal transduction pathways involving the G protein coupled receptors, the CAMP signal pathway and the phosphatidyl inositol signal pathway. So first, let us learn about the CAMP signal pathway. So CAMP stands for cyclic adenosine 3,5 monophosphate. So this transduction pathway regulates numerous cellular and physiological processes such as carbohydrate metabolism, gene expression, heart contraction, folliculogenesis, cystic fibrosis, transmembrane conductance regulator, which is known as CFTR activity. So when a person unexpectedly comes face to face with a tiger, his or her body quickly shunts the blood away from the skin and digestive system towards the muscles. So the heart starts beating faster. The liver will release glucose molecule, which actually provides the fuel required for what is known as flight or flight response. Is it not true? If you come across a wild animal, you have to either fight with the animal or you have to fly away from that place. So this kind of response is known as fight or flight response. So in this fight or flight response, the adrenal glands releases a hormone called as epinephrine. So this serves as a signal within the body. So cells like liver cells can detect these signals and they process the signal and respond to it. So the entire sequence from signal reception to the response is what is called as signal transduction pathway. So this is the outline of the CAMP signaling pathway, that is cyclic adenosine monophosphate signal pathway. Here you can see the ligand. This will come and bind to a G protein receptor, and you see the series of actions that is happening, and finally what happens. So let us now learn about this in detail. So the first step in epinephrine signaling. Okay. The first step in epinephrine signaling occurs when the hormone binds to an epinephrine receptor on the cell surface. So the hormone triggers the receptor to change its shape and convert the receptor to its active form. So the activated receptor triggers a cascade of events within the cell, beginning with the activation of a G protein. So the G protein binds to the activated receptor, releases GDP and takes up a molecule of GTP. Here you can see the ligand. So this is nothing but the hormone epinephrine in this case. So this will come and bind to the G protein coupled receptor. So earlier the shape was this. Now it is undergoing transformational change and it gives away GDP and takes up another molecule of GTP. So after taking up the GTP, the G protein is released from the receptor and splits into two parts. Here you can see after the GTP is being picked up, the G alpha, G beta, and G gamma components get split. So after it splits into two parts, one part gets activated, and continues the signaling cascade. And soon the hormone also leaves the receptor and the receptor reverts to its inactive form. So here the hormone is nothing but the epinephrine. So first it will go and bind to the G protein or the G protein coupled receptor. It brings about a conformational change. It takes up a molecule of energy and releases a molecule of GTP. And this will bring about uh, the release of the G protein from the receptor. Here you can see the G protein receptor has three components, G alpha, G beta, and G gamma. So now it splits into two parts, G alpha as one part and G beta and G gamma as another part. So G alpha is the active part. So this will take up another molecule of GTP, and then it continues the signaling cascade. So here the G protein activates an enzyme called as adenylyl cyclase. Here you can see adenylyl cyclase. So when it is activated, it converts a large number of ATP molecules into signaling molecules. So it will start taking up ATP and convert it into cyclic AMP. So this cyclic AMP carries the message of the first messenger, that is epinephrine, into the cell. So CAMP is referred to as second messenger. Since it is carrying the information that is carried by epinephrine, it is now called as the second messenger. In response to an internal timer, the G protein soon inactivates itself by cleaving GTP and the subunits reassociate. So here you can see once it is turning on adenylyl cyclase and converting ATP into CAMP, it gets deactivated and it reunites with its earlier components G beta and G gamma by cleaving one molecule of GTP, and again it dissociates from adenylyl cyclase. So with the protein no longer attached, the even adenylyl cyclase will turn off and can no longer convert ATP into CMP. So as long as this G alpha is attached to adenylyl cyclase, it can convert ATP into cyclic AMP. Once this G alpha component itself reassociate with the G beta and G gamma, even the adenylyl cyclase will turn off. Now the cyclic AMP molecules, which is produced by the adenylyl cyclase, now they continue the signaling cascade by binding to a type of enzyme called protein kinase A. So this binding triggers protein kinase A to separate into subunits, two of which are already active catalytically. So here you can see the protein kinase A. This is the inactive heterotetramer, but it has subunits, R subunit, okay, which is actually active. The activated protein kinase A subunits perform chemical reaction, which they add phosphate groups to another type of enzyme called phosphoryl kinase. The addition of phosphate groups activates the phosphoryl kinase. So along with protein kinase, there's a phosphoryl kinase. So by addition of phosphate group to this kinase, activates this particular enzyme, phosphoryl kinase. So once this phosphoryl kinase phosphorylates another enzyme, the cascade called glycogen phosphorylase. When phosphorylated, this enzyme also becomes activated. So in its activated state, glycogen phosphorylase produces the cellular response to epinephrine. So glycogen phosphorylase breaks down glycogen to its component glucose molecules, and during the process, the enzyme adds a phosphate group to the end of each glucose subunits. So there's one more enzyme which one add ಮಾಡ್ತೀರಾ ಇನ್ನೊಂದು remove ಮಾಡುತ್ತೆ. So this enzyme removes the phosphate group from the glucose molecules. So without the phosphate groups, now glucose molecule can be transported across the plasma membrane of the cell. Once outside the cell, the glucose enters the bloodstream. It is taken up by other cells and is used as a key component of epinephrine-induced fight or flight response. So signal transduction pathways allow cells to respond to environmental signals. In the majority of signal transduction pathways, a signal is amplified such that most steps produce a large number of activated components than the previous step. So signal amplification, for example, results in a liver cell releasing many glucose molecules after detecting just a single molecule of epinephrine. So signal amplification can occur at many points. For example, as long as epinephrine remains bound to a receptor, the receptor can activate a succession of G proteins. In addition, each adenylyl cyclase enzyme can convert numerous ATPs into cyclic AMP molecules. Other activated enzymes in the pathway can also continually catalyze reactions. The G protein, in contrast, activates just a single adenylyl cyclase enzyme and must remain attached to it in order for adenylyl cyclase to remain activated. Termination of the cellular response is as important as its initiation. In order for a cell to respond only when a signal is present, the many players in pathway have to be regulated so that they're activated only for a shorter period of time. Next is the phosphatidylinositol signal pathway. So besides epinephrine signaling, another important signal transduction pathway that uses GPCR is the phosphoinositide signal pathway. It is also known as phosphoinositide cascade. To give a particular example, we all know about this angiotensin 2 receptor, which is in our own body, and this receptor, angiotensin receptor, uses this pathway to regulate the blood pressure in the cardiovascular system in the heart. This pathway begins with the binding of the primary messenger to the GPCR. So this binding induces conformational changes that cause the G protein to expel GDP and bind a GDP. So here you can see the receptor, angiotensin receptor, is receiving the signal because the moment it binds with the primary messenger to GPCR, there's a conformational change and that releases GDP and it will bind to a GTP. So this causes the G protein to detach from the rest of the receptor and move on to and bind to a membrane-bound enzyme called as phospholipase C. PLC is nothing but phospholipase C. So it will go and bind to PLC. So upon binding to phospholipase C, the G protein stimulates it to begin breaking down of phosphatidylinositol 4,5 biphosphate, which we call it as PIP2, into two secondary messengers known as diacylglycerol and inositol 1,4,5 triphosphate, which is known as IP3. Here you can see PIP2 breaks into diacylglycerol and IP3. So this diacylglycerol is non-polar and it remains dissolved within the membrane of the cell. It moves along the membrane and binds onto the membrane-bound proteinase PKC, that is protein kinase C. IP3, whereas, is water soluble and moves across the cytoplasm and it binds to a ligand-gated calcium channel on the endoplasmic reticular membrane. Upon binding, it causes calcium ions to rush into the cytoplasm. So here the calcium does two things. First thing is it binds to the protein kinase C with the help of DHA and stimulates and activates the kinase. Second, the calcium binds to calmodulin, forming the calcium-calmodulin complex that then goes on to activate effector proteins and enzymes such as protein kinases. These kinases can then phosphorylate other proteins, enzymes, channels, and pumps that ultimately carry out cell processes that cause that particular physiological effect. That is, either it can increase the blood pressure or decrease the blood pressure. So to summarize, G protein coupled receptors are the biggest and most diversified collection of membrane receptors in eukaryotes. The beta adrenergic receptors that bind epinephrine is the best example for GPCR. An external signal such as ligand and another signal mediator activates the G protein coupled receptor. This causes a conformational shift in the receptor resulting in G protein activation. G proteins are then inactivated by RGS proteins, which are GTPase activating proteins. The CAMP signal pathway and the phosphatidylinositol signal pathway are the two main signal transduction pathways involving G protein linked receptors. Thank you.